these aren't even real strains. This is a terrible bit. What's going on, guys? My name is Voshko, and I'm here to teach, if you can call it that, if that's what you want to call it. I'm pretty passionate about science. I just graduated in May with a degree in biochemistry. I'm working in a neurology lab in NewsMedical. I write science articles on the side for a place called AzoLifeSciences. I have a periodic table, an archaic, kind of periodic table tattoo on my freaking arm. That should give you an idea of how nerdy I am. It's my shtick. And even though I do it professionally, I kind of want to do it for shits and gigs as well. And when I'm perusing through YouTube and I see people explain science or explain, one reaction or another, or if they're trying to educate a class or, tutor in some way or form, it's kind of monotone and drab.
Using up the, hot, insoluble cold.
Understand the
And depending on what kind of course you're looking at or what kind of video it is, what kind of topic it is, a lot of them are in Hindi or in Mandarin, or depending on how specialized, of course, the field is and where it's more prominent. So I felt like this could be a good medium if people are interested in, entertainment as well as learning something, because who wouldn't want to? Who doesn't like Hank Green? Who doesn't like Hank Green? So I'm here to address an English-speaking audience, I guess, about science in the wackiest, craziest possible way I can. So let's give it a shot. So, biotechnology.
Technology!
It's something that's kind of exploding right now because of your friendly neighborhood coronavirus. In every facet, in every field, it's nuts. It's going into transportation, industry, real estate, you name it. The whole shebang. And even though we see Uber Eats prices rocketing and plane tickets sky falling, there's a scientific front that's not being addressed all that much in the media, which I'm not about. So I feel like now would be a great opportunity to do so. So let's do this. We see over the course of history that technology sparks during times of crises. Like, for example, during the 2008 mortgage crisis, a lot more people were using a lot more companies, I should say, were using cloud-based systems and for security and accessibility and ease. And there was a trepidation in doing it, that people were really anxious and worried about doing it at first because it's such a new technology, but they found out it was so much better, so they started using it more. And it's because there are crises like that have a complete paradigm shift, and people start using a new technology or another. And after COVID-19 all but brought the world down to its knees, we saw an unparalleled level of biotechnology being upgraded and innovated. And inventions were predominantly in the fields of vaccinology and pharmacology. So, for example, a little while ago, the Zika vaccine came out, and it happened after 7 months of the virus already being present. So once we discovered the virus, it took 7 months to develop kind of a valid vaccine. With COVID-19, it took all of 42 days. And that's unprecedented. That's absolutely surreal. So let's talk about vaccines for a little bit so we have some understanding. So in vaccine development, there are generally 3 phases. First, there's the exploratory phase. This phase is where the vaccine is given to healthy volunteers to see if they, don't die, to see whether it's safe or not to use. And without going too much into detail about the biochemistry of it, the viral vector in question is going to induce an immune response, which your body is going to register and then use later on to combat the virus because it's already used to having fought it. Next comes the clinical development phase. This is where the clinical study is more or less expanded, and they're given to people with the medical condition that the vaccine is trying to treat. A small number. And the last phase is quality control, where it's given a couple thousand people, and if it works and we see results, then it's just shot off into the market, and there you go. And under past conditions, this would have taken a middling 15, 20 years, and recent breakthroughs have veered this course off completely. In the broad scheme of it, through the manipulation of genomic and episomal DNA, we can trigger an appropriate immune response via our antibodies. So current attenuated vaccines exist for the Rubella virus, measles, influenza, and a smattering of others. And to foster a live attenuated vaccine for COVID is kind of a big deal. The way this is performed is temperature-sensitive mutants are isolated from people that have contracted the vaccine and are replicated in low temperatures, 32 to 34 degrees Celsius. This is done so it doesn't impair proliferation of cells, so that the virus isn't completely dead, but it's alive enough to replicate and induce this immune response in people. But before all of this happens, the viral vector needs to be grown in vitro, not inside a human. It has to be grown in cell lines, for example. So in 293 T cells, HeLa cells, Bay1 cells, or animal models. And the way that they first weaken the virus before they distribute it amongst cell lines and populations is they weaken it through, there are several different processes. So there's acidification, where you're adding acid to it. There's heat, of course, because heat denatures DNA, or enough of it rather. They use radiation sometimes. And let's just keep in mind there's a bunch of different vaccines out there. There's conjugate vaccines, mRNA vaccines, protein subunit vaccines, toxicoid vaccines, attenuated vaccines, DNA vaccines, inactivated vaccines, synthetic peptide vaccines, and I could honestly go on. So bringing it back to the original point, what's caused this massive influx or spike in biotechnology, or virology really, is all these labs working in tandem on all these different methods of making a vaccine simultaneously. The most prominent vaccine stereotypes are Pfizer and Moderna, both of which are mRNA vaccines. And at least for now, mRNA vaccines and viral vectors only code for the spike protein. And this limits some ways the body's immune response because it's only focused on one viral antigen. But anyways, we've kind of covered how these vaccines are developed, but what's really cool is that it's tentacling out to other parts of science. So other branches of medicine like oncology and pharmacology are also being aided with these new biotechnological innovations. Really, if you want to think about it, mRNAs, or mRNA vaccines rather, are becoming the best and most versatile, actually, therapeutic out there. Because protocols and clinical testing associated with these new vaccines are being built around other diseases and ailments every single day. Did you know that there was no cure for rabies until RNA Active, which is a company, developed a vaccine for it using this kind of similar analog of technology? And viral vectors are now being used to cure stuff like prostate cancer and like 100 other different diseases. So for the rabies vaccine, for example, doses of 1,280 microliters are being given to these patients, and it's in phase 3 of clinical trials. And this could potentially save the lives of millions of people if, you know, an army of rabid squirrels comes and attacks you. Whatever, you know what I mean. This is a big deal. But it's also important to understand the weaknesses that come with these vaccines. Toxicity can be a point of contention because certain genetic vectors code for these unnatural peptides and proteins that we have in our body. And this can sometimes express some form of toxicity. Not much, but some. But these genomic analogs being used in these vaccines, quite frankly, kick ass. And there are people that take care of these toxicology reports and survey them and look them over and assay them to make sure that the stuff that's being pumped out to the general public is safe and valid. But these kinds of hurdles are natural for any medication, be it vaccine or otherwise, and it's something you're always going to have to succumb to or get past in lab. So yeah, this is my little rant. I hope that you learned a little bit more about vaccines and how they operate. I'm not sure if this is even going to be uploaded on YouTube or if this is, you know, just a moot point, but I had fun doing it. I don't know if this video is going to be like too much information or too little information, or maybe the information wasn't specific enough. I'm sure I'll get better at this as time goes along, but anyway, I hope you guys enjoyed, and I'll see you in the next video, maybe, who knows. Yeah, peace.